Well, thank you very much, and I appreciate the opportunity to talk about the process that I've been engaged in with the NCCN for the last, I guess, 15 or almost 20 years. So just as a way of background, and then we'll get into really the focus of the actual meeting, which is biomarker development and how we integrate those into actually practice utilization. The NCCN, I think everybody in the room is aware, is a sort of consortium of many of the leading NCI centers around the country, not all, but many of them, and they're geographically dispersed across the country. And really the effort of developing the guidelines was really after somewhat of the mandate and probably sessions that came out of this institute 25 years ago to try and develop statements that help physicians take care of patients in an optimal way and use evidence-based strategies to get the best of care to patients. And these were statements made in the early 1990s. So the guidelines, not only the NCCN, but many of the other ones that have made similar efforts, are really directed at a number of different rationales for undergoing the whole exercise. And that is to bring together experts who have acknowledged expertise in a particular area to look at the evidence, to do it in a transparent way, and to come up with objective decision-making that results in algorithms, which you're all familiar with, that take us from detection all the way through the full continuum of care for a patient with any specific disease. And obviously, if this is done in an appropriate way, they can serve as a way of monitoring or assessing quality of care. It also offers payers an opportunity to look at individual providers, centers, et cetera, and make an assessment about the appropriateness of care. And then, of course, within the NCCN guideline, these have served as educational tools not only for physicians, but also for patients and other caregivers. So within the exercise of developing guidelines, we like to think that every step along the way is clearly evidence-based. But as you know, from day-to-day practice, there are gaps in what we are able to provide based on evidence. And the level of evidence that really supports the things we do is sometimes not always what we would say the highest category, and I'll go over that in a moment. So as I said, we go through the process from detection all the way through end of life, and we also take into account things such as prevention strategies. But as you can see, there are gaps, and where those gaps exist, certainly within the context of guidelines, we try to bring the experts together and develop consensus where those gaps exist, meaning where the evidence isn't really as compelling as it might be in other steps along the algorithm. So with respect to the NCCN, I think, as you know, there's almost 50 multidisciplinary panels that are filled with anywhere from 25 to 30 individuals representing all the member institutions. And from those panels, we generate about 60 clinical guidelines. And these are available to patients, physicians, free of charge on the internet, and it oftentimes serves as the basis for insurance coverage depending on the situation involved. And although I could show you sort of a pathway for how we make decisions, for many of the large organ diseases, we meet face-to-face at least once a year, but it's actually a pretty dynamic process, and as information becomes available, say after an ASCO meeting or some high-profile publication, we will meet by teleconference, convene by teleconference, and we will look at the evidence in that setting, and then we will send our recommendations to the NCCN staff. It's ultimately reviewed by the panel chair and then disseminated back to the panel to make sure the decision-making that we had on the teleconference is not discordant with what we were talking about, and then ultimately it ends up online. And of course, we monitor subsequent data that may become available to make sure that we got it right. So as you know, there are different levels of evidence that we've utilized in this process. And it's not only the level of evidence, which I'll comment on in a moment, but it's also the quality, the extent of the evidence that we see in the literature. And by that, I mean, when we're thinking about quality, obviously what we view as a gold standard is a large, well-done, randomized clinical trial. And obviously a compilation of many, perhaps phase 2 trials may not reach the level of level 1 evidence, but it really is then getting into sort of a trend in the extent of the evidence that may fall short of category 1, but it's still fairly consistent. So we look at these different levels of evidence and we try and assign our recommendations along each node, regardless of the disease we're talking about, the level of evidence we assign to each recommendation we make. And I won't go through all of this, but as I suggested earlier, category 1 would really reflect pretty good consensus among all members on the committee. And it really reflects looking at high-level evidence that we would view as a well-done randomized trial. But for the most part, if you go across all guidelines, all diseases, you'll find that most of the recommendations or support for the recommendations falls within that category 2, and in fact 2A. And you'll see it, unless otherwise noted on the guidelines, that's generally what those guidelines reflect. And it's really category 3, which doesn't account for a lot of our recommendations, but it's clearly a controversial area, and it's so noted in the guidelines. And if you look across all guidelines and see where our recommendations lie, as you can see in red, the vast majority fall in that 2A, with about 6 to 10% of them being level 1 evidence. We just don't have randomized clinical trial evidence for every single node along the algorithms. But much of it is 2A, where there's a lot of consensus, even though it's not the absolute gold standard evidence. Now, with respect to the Compendium for Biomarkers, this was launched in 2012. And really, again, this was meant to segue very nicely with the development of the guidelines. So in all places where the guidelines had an opportunity to comment on a biomarker that was appropriate for making clinical decisions, whether it has to do with screening, diagnosis, monitoring, or providing predictive or prognostic information, this was incorporated into the guidelines, and the same levels of evidence that I mentioned earlier were assigned to the use of these biomarkers as they were to treatment decisions that we had elsewhere on the guideline. And these recommendations have been updated as new evidence becomes available. So very much the same as how we would approach therapeutic recommendations within the guideline. The same has been the exercise that we use for the biomarker compendium. And I should say that although we may recommend certain tests or define certain tests, we don't necessarily define the platform or the methodology that's used. That's really not the business we're in, but rather to look at the evidence that supports looking at each of these. So what would we consider a biomarker? It would be any measurable diagnostic indicator to use that we can assess risk or presence of disease, and this may look at specific genes or gene products. And again, these are put in the compendium, but not necessarily the exact assay or the exact methodology. So we stayed away from getting lost too much in the weeds of saying which is the best, very best test. As an examples of biomarkers in the guidelines, and I'll give a longer list that you can look at, but for risk assessment, obvious things like BRCA1 and 2 testing, PSA testing for screening and prostate cancer, BCR-ABL and leukemia, prediction HER2 status to determine whether a patient is a candidate for anti-HER2 therapy within the context of breast cancer, looking at CEA levels for colorectal cancer. We could go through a whole series of screenshots if one were to actually go onto the compendium site. There are many ways this tool could be used, and you can approach it from a disease-oriented approach where you can adjust the filters to what you're interested in. In the example of colon cancer, you could put that in, and it will look at each place in the guideline where there is a recommendation or suggestion that a biomarker can be used. And if you look at the bottom panel, you can see that different things, whether it's BRAF, CEA, et cetera, places where any of those types of markers have been alluded to and the level of evidence that supports them are outlined. Similarly, you can go the other direction and say, within the compendium, where is HER2 an appropriate thing to consider? And of course, we would think of breast cancer, but additionally, of course, there are certain subsets of gastrointestinal disease where this may be an appropriate marker and has some utility. So again, for the user end of this, you can change the filters and approach it in any way that is most useful to you. And of course, there are informational sheets that you can simply look at the role of, as an example, BRAF and melanoma, and it'll outline what the test is, what the marker is, where it's used, what the treatments that would be appropriate if that's expressed or not. So the criteria for clinical usefulness that we would use is that the biomarker affects treatment decisions. So somehow, is there evidence that's currently available that would suggest that we can actually employ this and have an outcome that's favorable? We can divide patients into relevant subgroups, and by extension, have different treatment decisions based on that subdivision. And again, as I said before, although we may suggest what biomarkers should be used, we don't necessarily say the tool or assay that must be employed to explore it. So within the compendium, just a list of things, there's a number of recommendations, obviously, with protein expression, translocation, mutations, chromosomal deletions, gene arrangements. And again, this is tied to a specific place within the compendium. And if you look at a more extensive list than I suggested earlier, you can look across the different algorithms, the different guidelines, and specific tests are suggested for specific diseases, which again may have some impact on treatment choices, specific targeted therapies, which has been the discussion yesterday as well. So finally, of course, there are concerns with developing this. We want to make sure that number one, by utilizing the compendium or having available to physicians the compendium and instruction about how to use it, that this can be employed for treatment decisions for optimal standard of care. But as clinical trials are evolving, we also want to be able to be using these tools as a way of entry or enriching clinical trials for certain types of patients where we may have tissue agnostic type of trials where we're looking at specific mutations that may cross a number of different diseases. Again, there is a lot of confusion among payers, perhaps clinicians as well, about which tests should be used. And you know, there's going to be a lot of discussion, and has been, about who's going to pay for which test should be utilized, what assay should be employed to assess a given patient. And there's obviously a proliferation of all these tests. What we tried to stay away from, and I think I probably said it 3 or 4 times now, is to designate the specific platform that should be used, but hopefully to encourage industry to have some standards about how things are being tested and also how things are being reported, so there can be a general consensus about how to optimally use the information. So I think I'm going to conclude there. My goal was simply to give a survey of the guideline process, and then how that segues into our more recent effort of using the compendium to complement the guidelines that we already have in place. Thank you.
I think since Bill, you have a little extra time, are there any questions directed just for Bill right now before we stop?
So we're in the era of electronic medical records and meaningful use, which includes requirements for clinical decision support infrastructure. So it seems like an ideal time for organizations that have focused on documents for the last 20 years to be thinking about algorithms. And thank you for calling them algorithms, right? Because they are kind of branching logic. To have an effort in electronic kind of computable versions of their guidelines. Is NCCN doing any of that?
We are, and there is an effort to actually integrate that into things like, as an example, Epic. And you know, we don't want it to be so cookbook that there is no decision making left for a physician, because it has to be individualized for care. But certainly the guidelines, and when you come to a certain place in the medical record, you may get a designation for things that could or should be done based on the situation the patient is currently in. Yeah. So I think we're moving in that direction. Obviously, to do that on a mass scale is no minor undertaking, but I think that is a goal that we're collectively trying to reach.
Yeah, excellent. And just even recognizing the phenotype and letting the practitioner know that there is a guideline.
Right, exactly.
One of the things we all struggle with is a concept of when does it become absolutely standard of care and when does it become something that is experimental. We talked about this yesterday, and how an idea that I introduced, and I'm Dane Dixon working with Palmetto. We talked about, you know, transitional markers. And we see that in certain guidelines there has been a not consistent adherence to levels of evidence when certain markers have been introduced. Is there something that is going to take place to try to establish guideline panel standardization to make sure that certain markers are introduced, that one guideline committee does not introduce something in a way that another guideline committee would say, hold on, that doesn't meet the levels of evidence. And then the second question, is there any thoughts on trying to identify markers where further information is absolutely necessary? Because one of the problems we see is that when it makes into the guidelines and then and we're still collecting research in some of these areas, that by putting in the guidelines, it immediately says in certain clinicians' minds that this is something is now standard, and they will no longer try to put those patients on clinical trials or try to collect more information regarding some of those markers.
So, that's a complicated question. In a global sense, yes, we'd like to have consistency across all the guidelines about how we approach anything, whether it's a therapeutic decision, whether it's inclusion of biomarker, and hopefully it's based on the same interpretation of level of evidence across the guidelines. There is realistically a tension that exists within different disease sites. I take care of patients principally with breast cancer, where there's lots and lots of different therapeutic options, whereas in other disease sites, they're much more limited. And I think the guidelines are all populated by experts that would be acknowledged by experts by anybody. And in those disease sites where there's limited options for patients, I wouldn't say regrettably, but I think the options being more limited, there's more a liberal, perhaps, interpretation of what we should be doing for patients when there's a paucity of options that they have. And in those situations, I think people are more willing to adopt strategies that have less support for them at the moment. I mean, ultimately, of course, we all want to exercise exactly the same interpretation of level of evidence. But I think for translating it to patient care in certain situations, we're a bit more liberal than perhaps across all the guidelines.
Great. Thank you very much. Okay, well, it's wonderful. We heard about the guidelines. Now we need to pay for them. And we're very fortunate to have Dr. Jennifer Mallon from WellPoint, where she's the Medical Director for Oncology Solutions and Innovation. And she's going to talk to us about treatment pathways. I know WellPoint made the headlines, I guess, around 6 months ago with a very, I would say, innovative incentive plan for how to get doctors to follow guidelines. So hopefully she'll talk to us a little bit about that too.